Welcome to Click for IB Biology. We're covering the topic of diversity of organisms, and here we're looking at the application of genomics into evolutionary relationships and indeed to genomics and health studies. There's a blank version of this presentation, a PDF is down in the description. There you'll also find linked in 10 questions that you can use to check your understanding of this particular part of the course, and there's the answers to consolidate your understanding. So let's get into the use of genomics in establishing evolutionary relationships and then take a look at the health application. So diagrammatically on the left-hand side here, I'm implying that there's a variety of species, there's a small selection, and we can see immediately that the diploid number is different from one another. And so this is the perhaps one of our first stopping points in looking at genetic diversity is the chromosome number. But of course, as was previously shown in other videos, there is appears to be no relationship between the genome size and complexity is not a relationship that can be seen. And this is particularly profound when comparing some of the sizes of smaller organisms such as Drosophila with human beings and of course with the flowering plants, showing their tremendously large chromosome numbers, largely as a consequence of polyploidy effects, which is one aspect of the genomic size. So in this genomic research, what has been done is that the DNA of organisms and indeed of their RNA is extracted, and the base sequence of this DNA and RNA is then sequenced into the sequences of the bases, which you know very well, A, T, G, and C. And then we begin identifying, for instance, within those, the number of genes. As we're doing so, we will also be able to compare the base sequence of those genes or sections of DNA. And we note that there are often large sections of conserved genes or conserved DNA, and these frequently correlate to genes which have been retained across many species, the greater diversity of organisms, such as the cytochromes, because they are critical in the functioning of the cell chemistry or critical in the development of the genes. The homeobox genes, of course, would be an example of the greatly conserved genes that you might find. But also, of course, as we move from one group to another, from one genus, family, or group to another, but then, of course, between species, as we start to see in the comparison of the base sequences that they have unique genes as well. And so there are techniques that we can do, and you will practice in your classes, where you can align homologous sequences of genes which are common to species, and to identify their conserved regions and where they have diversity. And so by doing so, we can then take that data and begin to construct phylogenetic trees. And these phylogenetic trees create a number of features for us. They show the classification based on DNA and RNA molecular information based, and that reflects their evolutionary relatedness in the structures that you can see. And so the one that I'm showing here, it will be familiar to you. I'm particularly familiar in this region here, where we have been looking recently at speciation within the great ape group. And so these diagrams reflect time in evolution. The nodes, which are here, reflect major branching and splitting events between those groups. And of course, in the study of any of these groups, what one would be doing is tracking back to try and find what the evidence was at the molecular level for these differences, but also at the phenotypic level and the gene level in terms of what this meant for the ecology and the diversity of these groups. Other applications of genomics are occurring in medicine today, where it's now possible for an individual to have their DNA extracted. It's a pretty straightforward process these days, and for sequencing to take place of their DNA. And this has now been accelerated by techniques known as next generation sequencing or high throughput sequencing. These are more contemporary methods which often employ robotics and artificial intelligence to handle the large quantities of data and information that are required for identification of genes and sequences. So through the identification of genetic sequences, it's now possible using this DNA to produce the diagnosis of a particular condition. Now, these diagnoses could be in real time. The person is actually suffering from a disease and one can find the genetic basis for that. Or it could be in a more predictive manner that one takes the DNA and through its analysis can predict that it's possible that this genetic disease may be triggered in life, and to take steps to prevent that from actually having or to mitigate the influence of the genetic influence on disease. And of course, this therefore leads to the great potential for the personalization of gene-based medicine for the individual, that your DNA sequences in their genome, in the total genome, are unique to you and give you a unique set of indicators of where your health may be promoted and enhanced. There are already around the world a number of major organizations, only two of which I selected from the data available. One of them is the Genomics England group, are working in conjunction with the National Health Service of the UK to enhance their portfolio of diagnoses of genetic diseases. This has actually been running for many years, but of course with these new technologies, the work can be greatly enhanced. And of course, you can also take a look at the Center for Genomic Medicine, which is a branch, I understand is a branch of the Harvard Medical School, and you can see the applications there of genomics in the treatment of disease, the diagnosis of the disease, and the creation of personalized medicine programs for individuals. The links to these are also down in the description for you to pursue, and I'm sure you can find others to represent the range globally. And so that brings us to the end of our videos on diversity in organisms. There's the chance to subscribe to the channel there, and the playlist as a reminder of all the videos that we have in this sequence.